MIP302. This is Catherine Kokinius, your Section 4 GTA, and I'm here to present you a little vlog video about our experiments. The purpose of these are optional, 100% optional. They're going to be a little bit silly sometimes, but also informative. I'll have like little pictures of my dog throughout, but just so you know, it's basically to kind of recap and talk through the theory behind these tests and maybe give you a few hints on how to do those questions on your post lab that are hopefully, this will be a little bit more interactive and should accompany reading the book. All the answers are in the book. These logs will not cover every single aspect of the book that is written in there, so I would highly recommend reading it. But myself and Laura, the other GTA for Section 5, will also be doing videos like these. I can say for, like I said, my own, I will provide some tips and things that might help with your post-lab questions, just things to kind of think through, and we'll draw on some fun whiteboard that I have here. So if you are down for that, continue watching. If this does not pertain to you, that is fine too. Cool, so let's start with Lab 9. So this is all filmed on my GoPro. So if you see a chunky finger in there, it is mine. Let's start off with the Ambach Tests. So they made this lovely, nice acronym. You can read more about what these tests are used for in your book on page 98. But let's go through each one. So the first one that we're going to go through is the eye indole test. So let's look at that. So the indole test is looking for an enzyme, and that's tryptophanase. The ACE indicates that it's an enzyme. So whenever you see that ending, you can recognize it's an enzyme. What happens is that enzyme can utilize tryptophan as its source of carbon and as an energy source. And that will catabolize, this enzyme here, will catabolize tryptophan into a product that we can detect indole once we add our reagents, our COVAX reagents. So the media that it's used to grow in is our tryptone broth. So another thing to note, and this is a 48-hour test. So it's incubated for 48 hours, and it tells us if this enzyme is present, if indole is present, which we should see by a red ring around the top. Here's that. I don't know how close I am, but hopefully enough to see. And if it's a negative for that particular enzyme, you will not see a ring at the top. No color change. Red, positive, no color change, negative. Okay. The next test is the M, which is methyl red. And this is also corresponds with our Voges-Prosker test here. So that's the V. So these two are related. So what we're looking for is, can this particular bacteria ferment glucose? And then the media is an MRVP broth, which has glucose in it, because we're trying to test if it can ferment glucose, and peptones, because we want them to still live, right? So you add your little bacteria guy into your tube, you incubate that for 48 hours, and then when you come back, you're going to split this solution to do two different tests with different components that you're going to be adding. So for the methyl red test, crazy enough, you're going to be adding a pH indicator, methyl red, which produces a red color. When it's in an acidic environment, it can also be dark orange, and that will be a positive MR test. If it's negative, it will be yellow or orange in a basic environment. So that's what this test will look like for positive and negative. For our VP part of our test, we'll be adding reagents. There it's A and B. And if it's positive, they'll be red in the upper portion of that solution. And if it's negative, there will be either no color change or green or copper color when these two reagents react. So either of those would be negative. The red layer at the upper portion would be positive. When doing questions on that talk about these tests, all of the ones that we've been talking about so far, you want to reference your book about the number of drops that are added and how long it's incubated for, because that could help you answering those questions. So make sure you look at the procedure even though we're not actually going to be doing it. So the next test we're going to talk about is our citrate test, which is right here, right? The C of our test. Okay. So our citrate test is on a slant. So this is like a solid. The other two were in bras. This is on a type of agar. Right? And so the agar name, the media type is here, Simon citrate agar. What we're looking for is, can this bacteria use citrate for a carbon source and ammonium ions as a nitrogen source? And we have a pH indicator, which is bromethyl blue. And we're looking for alkaline products. So she basic. She going to Starbucks, she's having a good time, drinking her frappuccino. So very, very basic. So if you're seeing it's going to be positive, it's going to turn blue and you're going to see growth on your slant. And it'll be a blue slant with growth on it. So the way that I remember this would be that, okay, so it's a basic environment, alkaline products. Most people don't want to be basic. The bacteria is kind of sad about being basic. Another way of saying sad is feeling blue. So it's basic, the bacteria is feeling blue. So if it's negative, there's going to be no growth and it will remain a green slant. So if there's no growth because it can't use citrate as a carbon source, it can't use the ammonium ions, so it's not going to live really. So, all right, here's our urease test. As you can see again, there's the ACE ending enzyme, right? We're looking for, does it have this particular enzyme here? The way that we do that is we grow it on a particular type of media called urea agar. This contains urea, glucose, and a pH indicator of phenol red. And this is a 48-hour test. So these appear on a slant, and the slant starts off as this peachy color. So a positive result is when it turns hot pink. That lets us know that it has this particular enzyme, that it used the urea that's in that media. A negative result typically is a peachy color, no color change, and no growth. It didn't ferment the glucose that was also in there, so no growth. It could also be a yellow looking slant. There's some growth. You can see right here, these dots indicate growth. And that means that it did ferment the glucose, but it is negative for what we're looking for right here, right? So as you can start to see a pattern here, we're looking at, can a bacteria do something? Does it have that enzyme? Does it not have that enzyme? Different ways to be able to differentiate them, which will be useful for your unknown project. So start kind of thinking about all of that and how these different elements can be put together to narrow down an investigation. This is our LDC and ODC decarboxylase tests. So they're very similar, so I put them on the same dry erase board.
And what I have here is, okay, what is it looking for? We're looking for a decarboxylase enzyme, so either our lysine or ornithine decarboxylase, so depending on which test you're doing. This is going to be an LDC broth or an ODC broth, like I said, depending on which test you're doing. And the pH indicator for both of these is going to be this guy here. And so what these actual tubes look like, it's a broth, so it's going to be a purple broth when you start. And there's a need for mineral oil. So what we're trying to do is we're trying to have an anaerobic environment so that mineral oil will help us do that. So if you have a positive test, what you're going to see is a turbid purple broth. So maybe I should draw some turbidity in there. That makes sense, right? Okay. A turbid purple broth, and that indicates that this is a basic condition. So in basic environments with this pH indicator, this will remain purple, and you will see growth. It will look foggy, turbid, right? And then depending on which test you did, if you did the LDC, this is your product. If you did the ODC, this is your product. Cadaverine and putrescine. Kind of sounds like a lot of death. So I might actually look those up and let you know after we talk about these what those are found in. And I think their names are self-explanatory, but just a little fun fact. All right, for a negative test, if it's yellow, that means it's an acidic environment, and that means that it can ferment glucose, which happens to also be in those broths, but it cannot use the LDC or ODC, the lysine or thiamine. So that's what that will tell us. That's a negative result. The other type of negative result is if it can't even ferment glucose, it's just going to be purple and it's going to have no growth inside. Okay? Okay, last one. So this is our Kia, our Klingler's iron auger. And what you're looking for here, there's a ton of stuff. I would highly recommend looking at page 100 and reading that section. So making sure that you also look at the time for this one. So this one is supposed to be only incubated for 18 to 24 hours. Some of the other ones, like I said, were more. We were doing 48, remember? So keep that in mind. And there is a nice, on the bottom of page 100, you can actually go over to it. Here's page 100. There's a nice little table right there that I've also drawn out here. Okay? So from this test, you can actually learn a lot of different things. You can learn if your bacteria can ferment glucose and or lactose, and if it's producing CO2 or it's producing hydrogen sulfide gas. And the pH indicator in this is phenol red. So what this is going to look like, it's going to be a slant. When it refers to the butt, it's talking about this bottom section of the slant. When it's talking about the slant itself, it's talking about the upper portion. And as you have all read, I'm sure, we are stabbing the bottom part of our slant here. So that is how that works. So in that chart, it talks about all these different outcomes you can see. So if it's red on the bottom here, that means negative for glucose fermentation, while yellow on that bottom butt part is positive for glucose fermentation. And if it's black, that means it's positive for glucose fermentation and for hydrogen sulfide gas formation. If the top part of the slant is red, it's negative for lactose fermentation. If it's yellow, it's positive for lactose fermentation. And if the auger is moved or cracked, it's positive for CO2 gas production. So when I'm talking about this, what you'll see is that the auger is actually moved up. So there might be even like an empty space in this bottom of this tube here. There'll be cracking in the auger. It's pretty obvious when you see it. So that is all the tests that we have for exercise 9. And as promised, I will have pictures and a little video of my doggo because you've sat through this whole video. Kudos to you. Are we gonna go outside? What? Outside? Hi! Hi! What is this? What is this? Sit. Lie down. Sit. Lie down. Crawl, 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 crawl. Good boy. Good boy. I'm so good all the time. Oh.
Fun fact of the day. So, putrescine and cadaverine are organic compounds that smell bad, that are involved in breaking down amino acids of both living and dead organisms. So, now you know. Say bye. Bye, everybody. Oh, he threw the ball. Okay, see you.